Hey guys, welcome to AP Biology or College Biology. We're going to be focusing in on Unit 4, Cell Communication and Cell Cycle, with more focus on cell cycle rather than cell communication. If you notice, earlier videos were all about cell communication, which is section 4.1 through 4.5. However, now we're pivoting and now we're focusing on the cell cycle portion of this unit. Before we get started, please note the disclaimer listed below, and let's get started. Okay, the big word is cell cycle. So what is the cell cycle? As you can see here, there's a picture that represents the cell cycle, and the cell cycle is all about how cells divide and become new cells and how they grow throughout their lifespan. So essentially what happens is a large chunk of a cell's life is in what's called interphase. And interphase is where the cell continues to grow and replicate and prepare to divide and become new cells. So this one cell will eventually become two new cells, but before it does, it has to make sure that it's ready to go. It has to make sure that this specific cell doesn't have any damages and has tissue repair and is fully able to grow so it doesn't pass on anything incorrect onto those new cells. So, what ends up happening is you have the cell cycle, which breaks down into two, maybe three parts. So the two major parts are interphase, and interphase includes G1, S, and G2. So G1, S, and G2. And with G1, S, and G2, if you look at the cycle here, you could see how big it really is. We're going to talk about G0 in either this video or a later video as well. But for now, you just need to understand that G1, S, and G2 are a large part of a cell's life. And it's really large because it takes a lot of time to ensure that cell is ready to conduct the smaller portion of the cell cycle, which is mitosis. Mitosis' main goal is to take one cell and produce two daughter cells, and that's where the M comes in, mitosis. And the two daughter cells have to be exactly the same. And that's the purpose of mitosis is to take this one cell and produce two daughter cells, we say, that are exactly the same. And with that, we don't want any incorrect genes that are passed on from the parent cell onto the daughter cells. Because of this, that is why interphase is so long, because it needs time to ensure that it's ready for mitosis, it's ready for the division portion. This usually occurs in eukaryotic cell cycles. They're highly regulated, and you want them to be regulated so that it doesn't lead to unregulated division or unregulated growth, which can lead to things like cancer. So regulation is really important through interphases checkpoints. The first checkpoint is called G1 or growth phase. This is where the cell essentially grows. And the next part is the S phase, which is DNA synthesis. So the cell will basically produce DNA for that cell and continue to grow into G2. So G2 is the second growth phase where it's preparing for that mitosis portion of the cell cycle. It essentially is preparing for mitosis so that way you can divide the nucleus, you can create one cell that divides into two daughter cells. So interphase is that major step that consists of G1, S, and G2. basically has checkpoints, and the checkpoints are there to ensure that we're ready to go. Like you want to think of an airport when you go through checkpoint to make sure that you have everything there before you go onto the plane and nothing is not okay. The same thing occurs during these checkpoints during G1, S, and G2. They're just checking to make sure that the cell is ready to go onto the next portion before dividing in mitosis. The cell cycle not only includes interphase, which remember is the most lengthy part of the cell cycle, and then we have mitosis, but we have this portion called cytokinesis. And the reason why it's not listed here is some people believe that mitosis and cytokinesis are so close together they're usually considered part of one another, but some cases they say cytokinesis is like you want to think the last step where the cell is finally dividing. So they sometimes put it together, sometimes don't, but just remember it's interphase, mitosis, and cytokinesis that are parts of the cell cycle. Interphase being the longest, where you're preparing for that growth, mitosis, where you're preparing for division of the cell, and cytokinesis when you're actually dividing the cell itself and including the cytoplasm in it, where you're dividing and essentially building a gap between the one cell to become two. And we'll explain more about this in just a second. So let's focus in more on interphase. So interphase, remember we said it is G1, S, and G2. So we know that the cell cycle's most largest part is interphase. And this is where we talk about the process of G1, which is characterized by that growth and cellular activity. The S phase, you want to think synthesis. Synthesis means creation, so you're creating DNA. And the DNA is going to be replicated. Remember, you have one cell and it needs to copy the DNA so that way it can get ready for mitosis to have two versions of the same DNA. So, for example, if you have DNA in one cell, you want to duplicate it so when it divides, each one will have a copy of that same DNA. So that's why you have DNA synthesis occurring in the S phase. Then we have G2, which is basically the final growth phase before mitosis, where you're preparing for that growth and you have the cell division almost ready to go. Now, we didn't talk about this, but there is a portion called G0, and it's when the cell is not really dividing, and it may or may not enter this part, but basically, it's just staying in G0 until we have the appropriate changes necessary for it to go into mitosis. So essentially what happens is you have cells that go through G1, S, and G2, and the cell is just quite not ready to go into mitosis. There could be something wrong with it, or maybe the cell itself isn't fully ready to go. So what ends up happening is the cell will remain in G0. And sometimes it will remain in G0 indefinitely. It will not continue on. Or what ends up happening is you have some cells that fix and will remove itself from G0 onto mitosis. So that's why you don't see G0 sometimes listed in the parts of the cell cycle, because it's a maybe not type of portion of the cell cycle. So just keep that in mind. As always, each of these portions have regulations and signals in place to ensure that we are stopping, making sure that everything's ready to go before proceeding forward to the next step of the cell cycle. What sometimes might occur is you have cell communication, where in mitosis, they can trigger adjacent cells to also undergo mitosis. You want to think of the fact that organs or parts of your cells in your body depend on other cells to tell it to grow or tell it to divide. And they could detect neighboring cells before making sure that they need to divide based on the density of nearby cells. So that's something to note, how cell cycle and cell communication sort of interlink together. So then we want to talk about mitosis itself. So when we're talking about mitosis, that is the smallest portion here. And when we're talking about mitosis, they have several steps. which you want to remember with PMAT. That's how I remember, PMAT. There are four main steps, sometimes five with that cytokinesis. But in terms of mitosis, remember, this is this part where you have one cell that will eventually divide into two cells that are exactly the same. But to do so, it has to go through four separate steps in between. And that includes PMAT: prophase, metaphase, anaphase, and telophase in that exact order. In prophase, what ends up happening is you have the nuclear envelope or membrane that disintegrates. So this nucleus right here will start to dissipate because, not this nucleus right here, sorry. This nucleus right here will start to dissipate. And the reason why that's doing that is because you have all your chromosomes and your DNA in there. And so you could see here during prophase, when you have the original cell and it's transitioning into prophase, you don't really have that circle anymore. And that's because it has to remove itself to ensure that you can count up the number of chromosomes, count up the number of DNA, count up that the chromosomes, which are, by the way, the chromosomes, when we talk about chromosomes, are what holds the DNA. So I might use the phrase chromosomes and DNA interchangeably, but it's safe to understand that DNA is contained in your chromosomes. So when you have a cell, you have a nucleus, and in that nucleus are going to be your chromosomes. The chromosomes will end up basically spreading out throughout the cell because they're no longer enclosed in that nucleus. And that essentially is prophase. It's getting ready for the next step, which is metaphase. And this is when the chromosomes itself are lining up at what's called the metaphase plate. And they're getting ready to spread to opposite sides of the cell, but not quite yet. And basically, what happens is the chromosomes will line up, and what they're doing when they're lining up is they have what's called spindle fibers that will attach to either side of these chromosomes. And the reason why is because it's sort of like a tug of war with the chromosomes where they're going to have to be pulled to opposite sides of the cell. So you can see here these little dotted lines are the spindle fibers. And as the chromosomes are lining up at the center, or what we call the equatorial plane, the chromosomes are getting ready to then go to the opposite sides of the cell, which is great. Because during anaphase, which is step number 3, they eventually take the chromosomes and you could see that they're split. So once a chromosome is like this, now they're split in this 2V-like shape, and they're getting ready to move to the opposite sides because those spindle fibers are moving them to the corners of the cell. So half of it is moving in this direction and the other half is moving in this direction. And that's called anaphase. This is all then going to turn into telophase, which is the last step, in which now we have an equal number of chromosomes in this potential cell and this potential cell. And then we have enough chromosomes that are exactly the same to eventually, as you can see, they're sort of bending in this shape here. That's called a cleavage furrow. And that cleavage furrow is getting ready to divide. When it's getting ready to divide, that's what cytokinesis steps in and says, okay, we're going to split these cells in half and go through our last step of cytokinesis. So cytokinesis is the last step that basically takes that little cleavage furrow in animal cells and separates them into two cells. Now, cytokinesis can occur in both plant and animal cells, and this is important to note. But if you remember in Unit 2, plant cells have what's called a cell wall alongside with the cell membrane. Animal cells don't have a cell wall. So it makes cytokinesis a tad bit different for plant cells versus animal cells. What happens is you have what's called the cell plate that's going to form in plant cells during cytokinesis, and it contains these rigid sugars such as cellulose that basically tends to help merge the existing cell walls together, and as it expands to eventually create two daughter cells. Eventually, what ends up happening is you need to create another cell wall or two cell walls to separate these two cells, and that's the difference in cytokinesis in plant cells versus animal cells, which develop what's called a cleavage furrow for animal cells, in which a little line dot will actually start developing, and Then allow the one cell turn into two. The cleavage furrow is basically going to be a groove, and you can see that little groove there that starts on the cell surface and sort of bends inward. You want to think squishing inward, and that leads to the cytoplasm eventually dividing into two. So you can see here cytokinesis is different in both plants and animal cells, which is a unique fact to remember in this case. Now it's quiz time. Take a moment. We're going to try to attempt the following question. Make sure you take a minute, pause the video, read the question, and then unpause once completed. Okay, great. Now that you're done reading the question, let's try to attempt this question together. It says, which of the following correctly describes the key difference between cytokinesis in plants and animal cells? And we just went over that. So if we were just quickly to go over the answers choices, the best one would be B. And I know that usually I go over each one, but I thought it best in this case because we just went over this, just to let you know the answer choice is B because we know it's different. It's not going to be that plant cells, so we can eliminate both answers. So answer choice C and D says both plant cells and animal cells form a cell plate, and both plants and animal cells use cleavage furs. So we can eliminate C and D based off this. So we're back to A and B. The reason why we like B versus A is, remember, animal cells have that cleavage furrow, while plant cells have a cell plate due to the cell wall. And because of this, answer choice B is the best answer. I hope this video helps. Make sure to hit that subscribe button. Make sure to review Unit 4 earlier videos. We have playlists and podcasts for up to this section so far. Make sure to hit that subscribe button for later videos just like this. Thank you.
Can you walk through the step of mitosis with this video? Try to see if you can find where prophase is. What about metaphase? Now find anaphase and telophase. Oh, do you see cytokinesis? There is that cleavage furrow. Do you see it? Now one cell became two.
Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams.